All right, today we are jumping headfirst into a real-life scientific detective story. I'm talking about a quest to solve one of biology's all-time biggest mysteries. And believe it or not, the whole thing hinges on one single, absolutely legendary experiment. So, get this. The experiment we're talking about has been called the most beautiful experiment in biology. I mean, that's a pretty bold claim, right? We think of experiments as, rigorous or maybe groundbreaking, but beautiful? That's a whole different level. It hints at something special, like a perfect, elegant dance between the question, the method, and the answer.
So what was this huge mystery? What question was so fundamental that solving it could be called beautiful? Well, let's dive in and find out.
Okay, let's set the scene. The year is 1953. Watson and Crick have just dropped a bombshell on the world, the double helix structure of DNA. It's iconic. And in their paper, they made this famous, kind of coy little statement that the structure itself suggested a possible copying mechanism. But see, that's all it was. A hint, a possibility. The real question, the big one, was still hanging in the air. How does DNA actually copy itself? I mean, every time a cell divides, it has to make a perfect copy. How does that happen? It would take another 5 years before a couple of scientists, Meselson and Franklin Stahl, decided to roll up their sleeves and crack the case.
You know, every good detective story needs a lineup of suspects, and for the mystery of DNA replication, there were 3 main theories on the table, each one a potential culprit. So let's bring them in for questioning.
Okay, suspect number 1, the conservative model. The idea here is pretty straightforward. The original parent DNA double helix stays completely untouched, totally intact. It basically acts like a template for a brand new, completely separate copy. Think of it like making a photocopy. You have your original and you have your new copy. They're 2 separate things. So after one round of replication, you'd have one DNA molecule that's all old and one that's all new.
Next in the lineup is suspect number 2, the dispersive model. Now, this one is, well, it's kind of messy. The theory goes that the original DNA molecule literally shatters into a bunch of fragments. Then, the cell uses these little bits and pieces to build 2 new DNA molecules. The result? You'd get 2 hybrid molecules that are a total patchwork, a jumble of old and new pieces all mixed together.
And our final suspect, number 3, is the semiconservative model. Now, this was the one that Watson and Crick themselves thought was probably right. It's just elegant. The idea is that the DNA double helix unwinds, kind of like unzipping a zipper, right down the middle. Each of those original single strands then acts as a template for a brand new strand to be built alongside it. So what do you end up with? 2 new DNA molecules, and each one is a perfect hybrid, half old, half new.
So, there you have it. Our 3 suspects are all lined up. But here's the million-dollar question. How on earth could you figure out which one is the right one? I mean, you can't just shrink down and watch DNA copy itself. This puzzle needed a truly brilliant, ingenious experiment. And that is exactly what our detectives, Meselson and Stahl, came up with. Their core idea was just so clever. They realized they needed a way to tag the original DNA so they could tell it apart from any newly made DNA. But they didn't use a dye or anything like that. Nope, they used weight. See, nitrogen is a key ingredient in DNA. So they used a heavy version of nitrogen, an isotope called nitrogen-15, to build the original DNA. Then any new DNA would be built using the regular, light nitrogen that's everywhere. Simple, but absolutely brilliant.
The experiment itself was just beautifully straightforward. Here's what they did. Step 1. They took some E. coli bacteria and grew them for a long time in a special soup that only contained that heavy nitrogen. This made sure that literally all the DNA in those bacteria was loaded up with the heavy stuff. Step 2. They scooped up those bacteria and plopped them into a new soup, one that only had normal light nitrogen. So from that moment on, any new DNA the bacteria made had to be built using the light nitrogen. Makes sense, right?
So the plan was to let the bacteria divide, and after each generation, they'd pull out the DNA and see how heavy it was. But how do you weigh DNA? Well, they used this really cool technique called density gradient centrifugation. All you really need to know is that they'd spin the DNA samples in a test tube at insane speeds. It works just like a centrifuge in a lab, or even a salad spinner. The heavier stuff gets forced down towards the bottom. So purely heavy DNA would sink low in the tube, and purely light DNA would float up higher. Okay, so the stage is set. The experiment is running, the suspects are waiting. Now all they had to do was wait and see what the evidence would tell them, generation by generation.
All right, first things first, let's look at our starting point. This is the DNA from generation 0. This is before they made the switch to the light nitrogen. And, yep, Exactly as you'd expect, there's just one single band, and it's sitting way down low in the tube. All of the DNA is heavy. This is our baseline.
Okay, now for the first big test. They let the bacteria divide just one time in that new light nitrogen soup. This was the moment of truth. What do you think they found? Would they see two bands, one heavy, one light, or just one band? And if so, where would it be?
And here it is, the first major clue. After that first round of replication, that original heavy band was gone, completely vanished. And in its place, a single band floating right in the middle, exactly halfway between where heavy DNA should be and where light DNA should be. What does that tell us? It means every single DNA molecule was a hybrid of heavy and light. Bam! Just like that, this one piece of evidence knocks the suspects right out of the running. Remember the conservative model? It predicted you'd have one all-heavy molecule and one all-light molecule. If that were true, we should have seen two separate bands. But we didn't. We saw just one. So goodbye, conservative model. You're officially off the suspect list.
Okay, so one suspect is down, but we still have two left. Both the semi-conservative model and the dispersive model could explain that single hybrid band. So the real clincher had to come from the next generation. The big question was, what would happen after the bacteria divided a second time?
And this is the smoking gun. After that second generation, they looked in the tube and saw two distinct bands. One of them was that same hybrid band right there in the middle. But now there was a brand new second band, and it was floating up in the light position.
This result was the nail in the coffin for the dispersive model. Think about it. If the DNA was just a patchwork of old and new, you'd never get a molecule that's purely light. The mix would just get more and more diluted, and you'd always have one single band that just gets a little bit lighter over time. But that's not what happened. The fact that a distinct, purely light band showed up, well, that was something the dispersive model just couldn't explain. And with that, suspect number 2 was out. So, with 2 suspects definitively ruled out, the evidence pointed in only one direction. There was only one culprit left standing. The case was closed. The evidence was absolutely undeniable. DNA replication isn't conservative, it isn't dispersive, it is semi-conservative. And this diagram from their actual paper shows you exactly why it works. It's beautiful. You see, in that second generation, those first hybrid molecules unzip. The original heavy strand gets a new light partner, making another hybrid molecule. That's our middle band. But the light strand from the first generation also gets a new light partner, and that's what creates a purely light DNA molecule. That's our top band. The theory predicted the evidence, and the evidence proved the theory. It was a perfect match.
So let's go all the way back to the beginning. Why is this called the most beautiful experiment in biology? Well, it's a few things, right? It's the sheer elegance of it all, using something as simple as weight to answer one of life's deepest questions. It's the clarity. The results weren't messy or ambiguous, they were crystal clear. It was also this powerful confirmation of what Watson and Crick had suspected all along. And maybe most importantly, it was the finality of it. This experiment didn't just add to the conversation, it ended the debate. It solved the mystery, once and for all.
And that really leaves us with something to think about, doesn't it? The Meselson-Stahl experiment showed us that science isn't just about getting the right answer. It's also about the creativity, the elegance, the artistry of how you get there. So the real question is, what is it that takes a scientific experiment from just being correct to being something truly beautiful?